what the name of the initiative was. You can tell us about the name of the initiative, Ken, but
yeah,
we're looking forward to hearing what you have to say. So please take it away. I think we might have to check.
Hopefully
we might just have to check we can see your presentation.
Hopefully I can share my screen with you.
So in the middle at the bottom, it should be a share button. There we go.
Okay, can you see my screen?
Yeah, is that PowerPoint or is it in PDF?
This is PowerPoint, yeah.
If you could just shove it onto, that's it, perfect. We can see it absolutely perfectly now. So Ken, over to you.
Okay, so this is going to be a fairly short presentation. It's going to be describing a project that we set up a number of years ago and is now coming to fruition. It's been titled OzBarley, from genome to phenome and back again. And this is a national program within Australia, supported by a large number of different organizations. So just to give you an outline, the OzBarley is a public resource capturing the diversity and breeding history of barley in Australia. So our plan is basically to try and identify all of the current varieties and look back through the pedigrees and actually identify all of the key varieties that have gone into making up the current elite varieties that we have it going at the moment. So within the project, we've collected pedigree data and genotypic data for approximately 220 elite lines making up the OzBarley core panel. And this panel is available to researchers to combine with their own phenotypic data to help fast track further gene discovery. So the OzBarley partnership, we have a number of funding bodies, the National Research Infrastructure for Australia, that's a bit of a mouthful, the Australian Research Data Comms, the Australian Plant Genomics Network, Bioplatforms Australia, and the Grains Research and Development Corporation. So these are our main funding bodies, but we also have a number of research partners. The main research partners are the University of Adelaide, CSIRO, the Australian Grain Genes Gene Bank, the James Hutton with our host, Robbie Waugh, and the AGRF, the Australian Genome Research Facility. And it's also a partnership with the main barley breeding programs within Australia, Australian Grain Technologies, Intergrain, and Secobra researchers. So there's a number of funding bodies, research organizations, and breeding programs involved in this. Now, our interest in barley is because it's one of the most important cereal crops in Australia. It's grown in almost all of the major states within Australia, except for the Northern Territory. We don't really get very high yields. Usually we get about 3 tons per hectare, but we have very large land masses. And we produced 7 million tons for export at a value of about 3.4 billion Australian dollars. And that's up on the average 10-year average from the 2010s, which is roughly about 6 million tons. Okay, so this project is basically to make data and germplasm available. And we've gone for what's called the FAIR data program. So this is an acronym which means findable, accessible, interoperable, and reusable. And the idea is that we are collecting germplasm that we make available to all the researchers that are interested in this. And we make all of the genotyping and phenotyping data that we've collected available for them. Now, initially, we started this project with an example cross of the Mundert-Kiel recombinant inbred population, which we had GBS data for and some phenotypic data. And we've used that as the standard population for us to build up our databases around. So we've chosen very early on to go with a local copy of Germinate. and we're hosting that locally on the Policy Data Portal. And we're also making available our data through Figshare. So, as I said, the whole idea behind the OzBarley is to collect a set of elite barley varieties. And we aimed for about 300, but we've ended up with about 220 lines. And for each of these lines, we've collected SNP chip data, so 40K SNP chip, RNA-seq data, and pedigree data. And we've combined that with phenotypic data, so growth analysis, spike and seed traits, and other traits that we've been able to do in the glasshouse. And we've also gone out into the field and we've run field trials where we've been phenotyping the material in the field. So in the field, we've collected height, head loss, spike and peduncle traits from the field, along with some basic soil microbiome data as well. Now, in addition to the elite varieties, we've also collected a set of 600 land races, and the idea was we were going to SNP chip them with exactly the same SNP chip as we did with the elite, so we'd have a comparable data set. Following the production of the collection, we've ended up with about 500 lines of land races. About half of them are winter and half of them are spring. And again, we've got basic measurements for that of height, peduncle, length, and row type. And all of that data so far has been collected in the glasshouse. We've not taken any of the land races out into the field yet. Now, the idea behind what we were doing is we wanted to have uniform germplasm that was fairly homozygous. So each of the OzBarley lines and each of the land races, we've actually taken through several rounds of single seed descent. So although the plants are derived from the original varieties, they are unique in themselves in that they are generally homozygous lines derived from single seed descent. Okay, so this is the basis behind the OzBarley panel. Originally, about 4 years ago, we made an open call to the community in Australia and asking them for materials of interest to be included in the panel. That includes all of the current varieties, around about that time, all of the major lines that were introduced from other countries that were really intrinsic to the breeding programs over the last 100 years. And as much as possible, we tried to source the lines that were in the pedigrees of all the lines that are the modern varieties. So a lot of the material in Australia tends to be early generation selection. So we have some issues with heterozygosity in some of the varieties and lots of biotypes that are present within the varieties. So we decided that we would try and make the material as uniform as possible by taking it through at least 2 rounds of single seed descent. So the breeding company, Australian Grain Technology, took that material and put it through their glasshouses and gave us back the material after 2 rounds of single seed descent. We then took that material, we bulked it up in the plant accelerator glasshouses in Adelaide, and we collected some basic phenotypic data on that in the plant accelerator. At the same time, we extracted DNA from that material and sent that off for 40K SNP chip data, SNP chip genotyping through one of the other barley breeding companies, Intergrain. And we also undertook RNA-seq. So our collaborators, who are partners in the project in CSIRO, extracted the RNA, and the Australian Grain Genome Research Facility undertook the RNA-seq work for us. We then took the pure material that we generated in the glasshouse and put it out into the field for bulking. The idea is that we wanted to have sufficient quantities to run numerous field trials. So we put that out to Western Australia, and Intergrain bulked that up for us and returned the seed to us at the end of 2022, in time for us to start field trials in 2023. Now, while this is all going on, we started looking at how we were going to set up our databases, data assembly with how we would work with the genotypic, the phenotypic data, and the Australian Plant Phenomics Network, which used to be the Australian it's Plant Accelerator, basically. They were being in charge of looking after all of our data analysis and data compilation and data release. Now, after we've generated all of this material, as I said, the material is derived from varieties, but it's not the same as the varieties because it's been through several rounds of single seed descent. So to make sure that future researchers can tie up the data with the material that we're working on, we've returned all of the single seed descent derived material to the Australian Grain Genes Collection. And they're going to curate and maintain that material for us in perpetuity and make that available to researchers on request. On site in Adelaide and in Western Australia, we've done the larger scale bulking, and we started taking that material out to field trials. So we have collected pedigree information, which surprisingly, given that most of this material was bred in Australia, there was very large gaps in our knowledge about the pedigrees of most of the lines that we had. And we really struggled at times to actually find the pedigrees. And we had to track down and ask a lot of information from a lot of our retired breeders to try and fill in as many gaps as possible. But what we've ended up with, we hopefully feel, is a good representation of the actual pedigrees as believed by the breeders. So, as I said, we undertook SNP genotyping. So that was on seed and leaf samples, and we used an Infinium 40K XT chip. And we also did RNA-seq at the same time of the same plants. And we found in a lot of cases, we actually ended up with data handling issues. And the SNP data and the RNA-seq data didn't actually match. So it took us probably the best part of a year to actually resolve all these issues and redo a lot of the genotyping and the RNA-seq work to actually get everything completely clear as to what we were working with. Other data that we've been generating, and I'll show you examples of that in a few minutes. We've generated X-ray CT imaging of harvested spikes, and we've collected growth and water use traits from controlled environment trials. We've got additional measurements that we've taken: height, tiller number, peduncle length, and flowering classifications. And all of that's been done in the glasshouse, but we've now put our field trial out in 23 and got the data back for that now. So we've got data collected for really what are just basic traits: height, head loss. Like I said, we collected spikes for X-ray analysis, and we also looked at the soil microbiome in one of the trials that was run out at Roseworthy, the university farm. Okay, so as I said, it was our plan to collate all the data and make it available in a single database. And we've been working with the James Hutton to work on the Germinate. So we've actually established a copy of Germinate here as a standalone version, which is hosted on one of the supercomputers based in Australia. And we're using Germinate as our data portal. And this is online, and that's freely available for everyone, I believe. There is a simple way of finding it just by Googling it. You should be able to find the copy and access to the data. The reason for choosing Germinate was its flexibility. And really, we wanted a system where we could add new data in future and really be able to expand and integrate the data from other sources. So the idea behind this is that we're doing the basic preliminary work where we do the genotyping, the phenotyping of basic traits. And this is going to be available to everyone that wants access to it on an ongoing basis. But what we would like to do is request that within 12 months of generating further data on the OzBarley collection, that it's actually returned to us and we can incorporate it into our database and make it publicly available. There's no obligation on people to do this, but we really would like to try and expand as much as possible of the phenotyping information that is being collected. Okay, so like I said, we looked at tracing the genetics of the Australian barley breeding. We started off with almost 300 varieties and accessions. We narrowed that down to 272, which we could actually get our hands on. And then we've put them into single seed descent to purify them as much as possible. And currently we have 223 accessions in the data set. We actually, not so much lost, but we weren't able to get several lines through the single seed descent process. And we've gone back and we're repeating that. But currently we have 223 and we hope to expand all of the data that we have available up to the full 273 as we go through the project. So we built a pedigree based on historical data, and you can see a representation of that here. And we also used the SNP data to actually build a pedigree network as well based on the 40K data. What we were expecting and what we actually saw was, although they're very similar, they're not identical. And it's not really unusual for us to find that. There's a lot of historical errors have gone on in making the varieties and recording the material that's gone into the pedigrees. So we didn't expect the pedigrees to match up exactly, but there is good correlation between what we see with the genotypic data and the historical pedigree data. Okay, so as well as the 40K SNP chip, we actually then did RNA-seq data as well. So CSIRO, Jess Hayes, CSIRO did RNA isolation on 4 days post-germination seedlings, and then sent that RNA to the Australian Genome Research Facility in Adelaide for sequencing. And we've got the data from that. And we wanted to combine that with the SNP data that we've got from the SNP chip. So first off, we actually looked at the RNA-seq, and we mapped that all back to the Morix reference genome assembly. So we used star alignment of the RNA-seq data with the Morix reference genome assembly, and Morix was included in the collection, so we actually had very good correlation with Morix. And you can see that as we go through, we've got less and less correlation as we go to CM72. But given the differences in the material that we've got, that's not really unsurprising. So looking at the RNA-seq data, we were particularly interested in using this to identify the transcripts and identifying SNPs within the transcripts. And this just is an example of some of the accessions. We've got Abyssinian, Gallian, Wimmera, and CM72 aligned to Morix. And we've identified large numbers of SNPs in each of these accessions. Zooming out a bit, we also found that there was quite a lot of indels there as well. And this is just an example of a 10 base pair deletion in Abyssinian. and gallium compared to the others. So there's lots of variation within our material. So looking at the 40K SNP chip data, we ended up with approximately 11,000 usable SNPs. But when we include the RNA-seq SNPs, we get an additional 110,000 SNPs, including the indels and presence-absence data, present-absence variants. So the SNP, the RNA-seq data has contributed enormously to the coverage that we've got over the genome of all these accessions. So, as I said, we started collecting phenotypic data, and we started off, first of all, doing this in the plant accelerator in the glass houses there. And we recorded grain size and grain shape. And we had an experiment where we actually looked at tiller number. Phil Brewer, who used to be here, is now in Queensland, was interested in using this material to look at tiller number and correlating that to strigolactone expression within the material. So all of this phenotypic data is being collected and put into the Germinate database and made available. We also have collected X-ray CT data on grain number and grain spike. And these are some images that we've collected. So across the population, we've been able to collect, identify the seed per spike. And this is just an example of the isolated heads that we've used for the CT scanning. In the plant accelerator, we're able to look at about 8,000 samples a week, which is roughly what we could do manually as well. But we get some lovely images that we can include in our database from this as well. Okay, so we've grown the material in the high throughput smart houses. And this is basically an automated system. Each of the plants are grown in a pot that's mounted on a conveyor belt. And once a day, they come through the smart house analysis system, where it's basically a Lemnitech system, where they're watered and they're scanned. So we get pictures of them, each plant photographed once a day over a period of about 120 days. So basically from germination straight through to maturity. And we can compile all these images together to actually make a video. And this, hopefully you can see, is a video of 4 of the OzBarley accessions growing over 120 days. This is vision captured daily by the Lemnitech. And we just basically used the image analysis software to stitch this all together. And this also allows us to calculate biomass and leaf area measurements as we're going through. Okay, so using this data, we were able to calculate growth curves, projected shoot area over time, and plot that out. And all of this information is being uploaded to Germinate. And we've also collected such phenotypic data, such as 2-row versus 6-row. And we've started undertaking GWAS analysis on some of these traits. So looking at the most likely candidates, so we're looking at the VRS and the Alice per se genes and early flowering genes. And we found associations pretty much where we'd expect them, which confirms that everything's going well. We've also looked at water use efficiency. And we found a single locus on chromosome 5 that accounts for 50% of the genetic variance. So we've looked at both the SNPs, RNA-seq SNPs and the SNP chips, but we also looked at variances in gene expression to generate associations. And last year in Roseworthy at the University Farm, we actually put out the 3 replicated trials of the material, and we went out and pulled up individual plants and extracted the DNA from the soil and did soil microbiome analysis. And likewise, we've undertaken GWAS on that, and we were trying to basically replicate work that was published previously, and we've included some of Klaus Pullen's Hebe lines as controls, and we've basically been able to identify genetic variants associated with the relative abundance of the microbiome genera. So the operational taxonomic units as measured by the abundance of the 16S RNA genes. So building on this work, this year we've got another field trial going ahead, but we were also taking material from the OzBarley into the glasshouse, and we're looking at variation in the application of levels of phosphate and nitrate in the glasshouse to see the effects on the OzBarley sets from the different levels of phosphate and nitrate, and looking to see if we can identify genetic loci associated with the control of the soil microbiome. So where are we at? We started off with the Mundar-Kiel data, GBS data, and some phenotypic data to go with that. We've now got the OzBarley Elite panel, so 220 lines of that. We've got SNP-CHIP and RNA-seq data. We've got the pedigree data. In the glasshouse, we've got growth analysis, spike, seed traits, manual traits. In the field, we've scored head loss, height, spike, and peduncle traits, as well as the microbiome. And we've also been building up our OzBarley landrace panel. We've got just around 500 of them. We've not taken them into the field, but we've grown them in the glasshouse. We've looked at the 40K SNP-CHIP with that material, and we've collected basic measurements for that as well. And we're returning all of our material now to the Australian Grain Genes collection, so that material will be available to everyone going forward. And we've also built our Germinate database and had our data kept here on a local variant of that on the POSI database. We've also uploaded a lot of our data into Figshare, and that's going to be available very shortly. So 2023, I said we had material out at the Roseworthy field trial, and this is just a picture of the field trial. And this is the landrace material. We actually have selected roughly half of the landraces as winter and half as spring. And believe it or not, we actually selected for as many of them to be as short as possible. But unfortunately, the passport data probably wasn't as accurate as we would have liked. And we've got some material that's more than 2 meters tall. Well, a lot of it's a lot more than 2 meters tall. So it's very difficult to work with in the glasshouse. And I don't think under Australian conditions we'll be putting it out in the field anytime soon. So what's next? As I said, we're depositing the seed back to AGT for distribution, and that's going to be available for anyone that wishes to request that. We made an open access call in, early on in this year for the field trials that we're running this year. We're running field trials in Western Australia, South Australia, and New South Wales, and we're providing travel funds through the GRDC or from the GRDC for early career researchers to go out and collect phenotypic data on their favorite traits at any of these sites. Some people are actually opting to go to all 3 different sites and generate data. So we'll have a nice set of data, phenotypic data collected later on this year. So after the initial analysis and QC of the key traits. All the data will be published. We're really keen that everything that we do is going to be published and freely available for anyone that wants access to it. And we're also developing visualization software to look at the data. And this is an app that's been developed by CSIRO. It's currently called the Shiny app, but that's just the in-house name. It will be called something else. And that is being funded by Platforms Australia and the Australian Plant Phenomics Network. They're investing directly into the OzBarley program to provide access to this Shiny app. We're hoping that as the project develops and matures, researchers will take this material and we'll invite them to contribute their own data in the future back. And that'll hopefully be available to everyone. As I said, there's no obligation on people to do that, but we're really hoping that people will join with the program and actually contribute their data back to us, and we'll make it available for everyone. There's scope to expand OzBarley. We're approaching the funding bodies for further funds. We want to do more with the land races. It'd be nice to do some RNA-seq with the land races. Basically, we see the land races as a source of material for allele mining, so people can actually go in and mine that material for their favorite genes and look at different alleles that are present there. And over this year, we are hopefully collecting additional phenotypic data from the field trials for a number of specific traits. And I'd just like to wrap that up and acknowledge the people that actually did the work. So Uta and Elena have really looked after all of the bioinformatics and the database curation. Julian has been in charge of the FAIR data and the data management. George has been involved in the data publication and setting up our installation of Germinate on Policy. And Pooja has been really key in collecting a lot of the data in the glass houses. And I'd also like to just mention that the PI for this whole program is Bettina Berger, who's based at the Australian Plant Phenomics Network, the Plant Accelerator in Adelaide. Okay, and as you can see in the picture, this is our WAIT 100th anniversary, and we've released several batches of whiskey to celebrate that. So it's good to see barley's going to good use in Australia. Any questions?
Sorry, I was muted there. I just wanted to say thank you. Thanks so much, Ken. Yeah, are there any questions? There are some questions in the chat. At least I've got a question in the chat. I'm just going to read it to you, Ken, so you don't have to read it. And I said, basically, I'm impressed by the fact that you managed to bring all of the listed academics, the funding organisations and the breeders together to support the OzBarley project. The question is, are the breeders using the data or the germplasm collection? Are they enthusiastic about the work? And have others not originally involved in the project requested access to the OzBarley data or the germplasm?
The answer,
you can reread it. You can reread the question in the chat if you like, just because it was quite.
That's all right. The answer is the breeders have been very involved all the way through this, and I mean they've been supplying access to their facilities to get this off the ground. And they've all been out to look at the field trials as well. So AGT basically did all the single seed descent material for us, and Integrain did, it was their chip, the SNP chip that we used for the genotyping. And basically without access to their knowledge of the pedigrees, we would never have been able to get much of this done. We see them as key partners. As for them using it, don't know that they are actually using it at the moment. We're still very much in the early stages of developing it. We have a number of projects with the breeding programs that use this material, so there are collaborations that have been established with them. And this year we have field trials in Western Australia. We have them in Sydney, and we have them in Adelaide. And one of the things that we were lucky enough to do was get funding from the GRDC to actually allow travel to each of these field trial sites. So we had an open call for scientists to request funds to actually travel to these sites and the collective phenotypic data for their favorite traits. And this was hugely oversubscribed, but we tried to give as many people an opportunity to go out there as we could. And certainly all of the people in Adelaide are going out to the Roseworthy trials and looking at that material here. I imagine similar, University of Sydney and CSIRO looking at the trials where they are. And Western Australia, they've got easy access to the trials where they are. So we've tried to make it as open and available to everyone as possible.
Sounds great. And we have other comments from Pierre. Pierre, do you want me just to ask this for you? I will do, because Pierre's probably muted.
Go ahead, Robbie. Yeah.
Okay, so Pierre says, thanks, Ken, for a nice presentation. He's wondering whether you've tested your material, including land races, for the main barley diseases.
No, we have not. We're limited to what we can do with our funding. So our main idea is to establish this as a resource and then make it available to other people to do what they want with. So this material is available to all our partner pathology groups across Australia, and I'm not aware that any have actually requested access to it, but they're certainly aware that it exists. And as a rule, all of this material will have been through many, many disease nurseries. So they would probably have much of that information already. I know that from a previous GRDC program that we had, working with the pathology groups, nearly all of these lines have featured in biparental populations that we've made previously to examine the major diseases in Australia.
Thank you. There's another couple of questions in the chat before I get to Sarah. James asks, do you think the future for Australian barley varieties are mainly probably adapted to very local conditions, or do you use for pan-continental varieties with wide-ranging ability to thrive? James, if I got that wrong, please open your mic and tell me what I got wrong.
No, that was good, Robbie. Thanks.
Yeah, look, we constantly are bringing in material from overseas, and some of the best performing lines within Australia are European lines, or have been European lines. Nearly all of our breeding companies are international breeding companies, so they have access to international germplasm to use at any time they wish. So we have fairly adapted material for the Australian conditions, but when we've looked at the material in the past, there's a great similarity between all of our material and European material. Even although the conditions are different, the genetics are very similar.
Ken, Misha's asking about the 40K chip. Is it customized for this project? And is it mainly aimed at Australian germplasm?
Yes, it is customized. It's a custom chip. It was developed by Intergrain and DPI Victoria, and it's used as a core part of their breeding program. Over the last 20 years, the breeding companies have been working with the breeding researchers developing molecular markers for a lot of the traits. And we've been heavily involved in the disease resistance traits in Adelaide. And we've provided gene sequences and marker sequences linked to these traits. And these have been incorporated in a large part within the 40K SNP chip. So all of the traits that they use for selection are in there already, plus a lot of other stuff that we're not entirely sure what it is that's in there.
Brilliant. So you've partially answered Pierre's question. So you've not scored them for disease resistance, but you've scored them for markers that are diagnostic for certain types of diseases. Okay. Is that fair?
So one of the interesting things about this collection is that the majority of the lines that are in there have been part of our winter cereal molecular marker program previously, which was a collaboration with the pathology groups all around Australia. So we have actually got biparental populations derived from the majority of the lines that are incorporated in the OzBarley collection. And these have been extensively looked at for disease in all of the different regions of Australia. So we have a huge amount of disease information and market trait associations that we know of that exist within this germplasm collection.
Brilliant. Sarah, you had a question.
Yeah, it's more just interest. You mentioned a phenotype of head loss that you were scoring. And is that head loss that would come from like snapping at the base of the spike, or is it head loss kind of from any cause?
This is work that Matt Tucker has undertaken.
Oh, okay, yeah.
So this is his area of expertise. My understanding is it is basically snapping off of the head.
Yeah. And is it there? Do you see a wide range? I mean, I can always ask Matt, but I'm just, what's your?
Sorry, I didn't catch that.
Is it quite a wide range of that phenotype? Like, do you see that some lines are consistent snappers and others aren't, or is it?
Yes, that's very much the case. And Matt's basically trying to determine the genetic control of that.
Aye. Interesting. Well, thank you very much. Sorry, I've got a dash, but that was a really lovely talk. Thank you. Look forward to seeing you in person.
So for those still around, Sal N, don't know what the N stands for, but Sal, thank you for writing the comments. And the Infinium 40K chip's also being used to genotype international germplasm and is being used to determine diversity across a broad range of germplasm, not just Australian genotypes. So I have a question coming back on that, Sal. What's the correlation between the SNPs on the 40K SNP chip and the SNPs on the widely available 50K chip that was developed by Misha Bayer, who's in the audience?
Ellie Norton here. I'm with the AGG. Can you hear me?
Yeah. How many SNPs are there in common?
I couldn't answer that. I know that there's been a lot of work in the development of the chip to make sure that there's correlation and good overlap with a range of different platforms used globally. But that, yeah, I can't answer that specifically.
All right, just wondered. Luke's got to go. I had a point, I guess, and a question. I'm not sure that. Well, I'll read the question. It's in the chat, but I don't think many groups consider the likelihood that genotypes get mixed up when populations are subsampled and distributed for multiple trials across multiple sites, and the data is collected and it might not match up. So you mentioned the difference you had between the SNPs that you got from RNA-seq data and from the SNP chip data. How extensive was that, Ken? Was it 10% or 20% or of the lines didn't match? I mean,
yeah, look, it was between 5 and 10%. It was significant.
All right.
We had a lot of problems, and it took us the best part of a year to go back and actually backtrack and check this out. There was an enormous amount of resources that we basically wasted on doing this.
Yeah.
But it needed to be done. So really, yeah, it was an issue.
Yeah, so I mean, the question really is, having gone through this process now, do you or the breeders or anybody in the Australian barley genetics community have a way of identifying what germplasm they're actually using when they're growing field trials in the field or doing experiments in the smart houses or glass houses or on plates? And the difficulty, of course, as you know, is that if you collect data on the wrong genotypes and then try to do genetic analysis with genotypic data that doesn't match the things that you're phenotyping, your analysis is going to be screwed up. So do you have a method for identifying, quick method, cheap method for identifying genotypes?
I don't know if there's a cheap method, but I certainly know that the breeders genotype all their crossing blocks, so they know what they're starting with.
Okay. Okay, I see Miriam Schreiber's in the audience as well. And I know personally that Miriam has had very similar experience trying to match up 50K SNP data with SNP chip data rather with RNA-seq data. And it's about the same frequency, about 10%, I reckon. Probably Miriam can comment if she wants. But it seems to be there seems to be this common ballpark figure of between 5 and 10% mix-ups in almost every genetic experiment that we've been associated with, which is a bit of a problem.
I had a quick look. I'm not quite sure what the percentage is, but the problem for us was also each location had a different mix-up, obviously. So it was a bit of a fun time to figure it out.
Can I just ask, it's 10 to 10 now. Does anybody else have any questions they want to ask? I can't see everybody, unfortunately, on the pictures phase page. So if you do, please just unmute and ask a question. No? And I have one question, Ken, again, just to finish this off then. I thought the data that you got from the Plant Accelerator looked fantastic. You know, and I guess most people online would think it looked fantastic and very high resolution and good quality. Have you managed to correlate the data you got from the Plant Accelerator with field data yet?
I don't know that it's actually been looked at yet. Unfortunately, there's no one online from Australia that can comment on that.
Yeah.
They've all gone home for their dinner. I don't know is the simple answer.
Yeah, okay. I mean, it's a question we always ask about controlled environment experiments now. So just a different environment, completely different environment from what you would get in the field. And we do wonder about the validity, basically, of it in terms of looking at phenotypes that are really usually expressed in the field and the field conditions. Listen, everybody, thank you very much for turning up. I realize that this was a different time from normal. So if you're overseas and it's late, I do thank you for turning up. I want to thank Ken especially for staying on and work. Must be 6:30 now there. So I really enjoyed it. Lovely, lovely presentation. And I'm kind of jealous about the fact that you've got this nice collection that everybody in Australia, I hope, will be using very soon. And also the database looks great, actually, having it in Germany. So it's something perhaps that we should aspire to as well. So well done. And thank you.
Couldn't have done it without you.
Yeah, sure. Thanks so much, Ken.
Okay, you're welcome.
One more round of applause.
Thank you.
Thanks, Ken. See you soon.